Hello, am I audible, students?
Yes, ma'am.
All right. Okay. Students, what were we doing in the previous class? Jyobita, can you check me what is the last thing you've written?
Types of allergy.
Types of
Allergy.
allergy. Okay. So did you people have any doubts from the previous topics that we had discussed?
No.
All right. Okay. Switch on your videos, both of you, and let's start today's class. Okay. So in the previous class, we were discussing about the allergy. Did I discuss anything further other than the allergy, immune system or anything?
Vaccination.
Vaccination has been discussed. And autoimmunity?
No.
Okay. Now see, we have seen what allergy is. It is a disorder of the immune system. Meaning, see, just like how there is a disease wherein a particular part of the body is affected by the pathogen, that's what a disease is. But when we talk about a disorder, the disorder is a different thing. It is like a generalized set of symptoms that a person appears, like feels or, undergoes, but it's not that only one part of the body gets affected as such. So we have discussed about the allergy. Let us discuss about autoimmunity. Now you people know that our immune system, it has this property wherein as soon as it will see any foreign particle, it will recognize the foreign particle and start to attack, right? So as to kill it. But when our immune system forgets to, or you can say fails to understand which one is the self body cell, which one is not, and instead of attacking a foreign particle, when our immune system starts attacking our own body cells, then that is called as autoimmunity. Understood? The property wherein the cell start, the whole entire immune system starts attacking its own cells. Some of the diseases that are considered as autoimmune are myasthenia gravis, Addison's disease, ulcerative colitis, and then there's rheumatoid arthritis, which is the most important amongst all of them. So this was autoimmunity. Now, when we discussed about it, now we moved on to the vaccination part, wherein we saw how during the vaccination, what is done basically? The you can say the dead organism, the part of the organism, or weakened organism is given or injected into the body, and as a result of that injection, the body or the immune system starts synthesizing antibodies against it. Now, when the pathogen actually enters our body to cause any, you can say, disease, then in that case, our body is ready to fight with it with the antibodies already made in the body. So, vaccination, my voice is clear, students? It's like clearly coming to all of you.
Yes, ma'am.
You're with us? Switch on your video. Now, talking about the different types of vaccination. So, there's first generation vaccination, second generation, and third generation. So when we talk about first generation, so this vaccine is prepared with the whole organism. The whole organism is taken. Either that organism is killed and whole, or that organism's pathogenicity has been taken, meaning it has been made weak, but still you're taking the whole entire organism. Okay, so that is like either you call it as weakened pathogen or you call it as attenuated pathogen. It's the same thing. Understood? So in this case, what happens is that the organism is given into the body, and this like examples of this kind of vaccine are cholera vaccine, typhoid vaccine. Then comes the second generation vaccine. So this one, the whole organism is not given, only the part of that organism, such as pathogenic protein. Those proteins that can cause infection, that can cause disease, that part is taken and that is injected into our body. So we call it as the second generation vaccine. And when the pathogen's DNA is just taken, nothing else, no protein, no the whole cell or anything of that sort. So that is a DNA vaccine, which is third generation vaccine, such as in the case of hepatitis. So these are the various types of vaccination that are there. Furthermore, talking about the symptoms of allergy. Symptoms of allergy, I think I have done, right? Symptoms and treatment of allergy. Yobita, have I done these two things?
Yes, ma'am. Till treatment.
Then comes the immune systems in our body. Now, our body obviously runs on the immune system because the immune system is specifically there in order to protect us from any kind of pathogen. Now, our human immune system has the lymphoid organs, has the tissues and cells and molecules such as antibodies. Now, lymphoid organs, in very, you can say, easy words, these are the organs where the lymphocytes, such as B lymphocytes, T lymphocytes, they are made and they are mature. These organs are what we call as lymphoid organs. So there are two types of lymphoid organs in our body. One is primary lymphoid organ and the other is secondary lymphoid organs. So primary lymphoid organs are the ones where the immature lymphocytes, they are going to differentiate. Differentiate means to divide, to increase in number, and to become the final, you can say, the lymphocyte. So such as bone marrow and thymus. Now you people know that bone marrow is that part of the body wherein all the blood cells are made and all the lymphocytes are made. Blood cell means RBCs, WBCs. These are made where? They are made in the bone marrow. And lymphocytes are, are also produced in the bone marrow. Lymphocytes such as B lymphocytes and T lymphocytes, both of them are produced in the bone marrow. The only thing is that the B lymphocytes, they are produced in the bone marrow as well as they are matured in the bone marrow. But when we talk about the thymus, now thymus, this is a lobed organ which is located near the heart, right? Just beneath the breastbone. And it is the site where the maturation of T lymphocytes take place. Meaning, the T lymphocytes, they are originally prepared in the bone marrow from scratch, but they are matured in the thymus. That is why they have gotten their name as T lymphocytes. And B lymphocytes have gotten their name because they mature and originate in the bone marrow itself. Now remember this one thing, that as the person increases in age, the size of the thymus, it decreases with age. And when the person reaches the puberty age, then the thymus has reduced to a very small size. Then comes your secondary lymphoid organs. Now, secondary lymphoid organs are not where the whole entire, you can say, lymphocytes will be produced, but they will help in the proliferation, right? To divide into more numbers. They'll help in the differentiation, and ultimately, these are the sites where the immune cells, they get activated. Okay, because our immune cells, they are going to produce any kind of effect only when they are activated, right? So they are activated to fight with the antigen. The secondary lymphoid organs, they trap the foreign particle. The thing they do is that they help the primary lymphoid organ products to produce a very well-renowned immune response. Is it clear?
Yes, ma'am.
Now, furthermore, talking about the secondary lymphoid organs and their examples. So there's spleen, there's lymph node, there's tonsil and appendix. These are all the different kinds of secondary lymphoid organ. Now, if you want to locate it in the diagram, just see this picture. See this picture? Here, you can see the location of all the various kinds of organs that are there. Jyobita, what are you writing? Hmm?
I was just marking.
Just marking what I'm teaching.
Yeah, in textbook, some points, important points.
But you pay attention here also, okay? Now see, when you're in the class over here, don't do any other work, okay? Try to learn what the teacher is teaching you. Then you're free to do whatever you want. So see here, this picture represents you the primary and the secondary lymphoid organs of our body. If you see here, the bone marrow, right? The part that is present in between the bone, that is the bone marrow where the B cells and T cells are produced. The thymus is present right beneath the breastbone, and this is where it is. Then the spleen, see this spleen is here, right? Then there are tonsils will be in the throat area. Then there are this thing, lymph nodes. Right, lymph nodes are circular structures that are responsible for trapping the this thing, any foreign particle. And at the end of the large intestine, like this part, not the end, the beginning part, this finger-like projection is what the appendix is. Have you people heard that many people get their appendix out? Why? Because it's not needed anymore, but it is also a, you can say, lymphoid organ. So when we talk about the first lymphoid organ, that is the spleen, so spleen is called as the graveyard of RBCs. Meaning the RBCs, once they have, completed their life cycle, they'll go to the spleen and they'll die over there. The spleen of the body, it acts like a filter, and whatever microorganisms are going to be there in the blood, these, like the spleen, will trap. Okay, and once it will trap it, the B cells, the T cells will come and kill it. Then comes the lymph nodes. Now the lymph nodes, they are small solid structures located in our lymphatic system. I believe you people remember that how there are two fluids in our body. One is the blood. The second is called as the tissue fluid, which is also known as the lymph. So this lymph is, or the lymph node is responsible for trapping the microorganism and antigens which are present in the lymph. Clear? Any doubts up until now, students? No doubts?
No, ma'am. No, ma'am.
Okay. Now, furthermore, what happens is that this, like these secondary lymphoid organs that are there, like I told you, they will increase our immunity. They are not the primary or the most important site as far as the immunity is concerned, but again, they're not also useless. Then comes MALT. This is very important to remember the full form of MALT, which stands for mucosa associated lymphoid tissue. This mud is actually the tissue, or you can say it constitutes 50% of the lymphoid tissue in human. Now, where is it associated? In all the mucosal layer of our body, such as the lining of the respiratory tract, the lining means the inner side. Okay, like towards the inner side, so the lining of the respiratory tract, the lining of the digestive tract, and also the urinogenital tract. These all areas have mucosa associated lymphoid tissues present. Any doubts up until now, students?
Um,
No, ma'am.
Oh, no doubts. Now moving forward. Okay, now we have the diseases to discuss. Systems completed. AIDS. Okay. So now we will discuss, like there are two diseases, there are major diseases that are given in your syllabus. One is AIDS and the second is cancer. Okay, both of these things we have to learn. So when we talk about AIDS, AIDS stands for acquired immunodeficiency syndrome. And the causal organism of this particular disease or syndrome is human immunovirus, that is HIV. Now, the nature of these viruses is that they are retroviruses. Do you people know anything about retroviruses?
Having RNA as the genome.
Okay, they have RNA as their genome. And one more thing makes them very, 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 very special. What is it? They have a very unique enzyme. Chalo, I've given you this much of hint. Now you tell. The presence of dash enzyme makes them very unique. What is the name of that enzyme? It was, I have taught you this thing in molecular basis chapter.
Reverse transcriptase.
Reverse transcriptase. Yobita, do you know what reverse transcriptase do? What does that enzyme do?
Forgot.
Hmm?
Forgot.
Forgot. How can you people forget? You shouldn't forget. See, retroviruses, they have that enzyme called as this thing, reverse transcriptase, which helps them. Jesslyn, do you know what the reverse transcriptase does? Tell me.
Converts RNA to DNA.
Very good. See, since this virus has RNA as its genome, like its genetic material is RNA, just like how our genetic material is DNA. Now, what this particular virus does is that using that reverse transcriptase enzyme, it synthesizes DNA from RNA. Normally, you people know that it is not possible to synthesize DNA from RNA. It is always this thing. The DNA to RNA to protein. That's how the molecules are synthesized. But in the case of HIV, which is a retrovirus, they can synthesize DNA from RNA. Normally, it's not possible. So, AIDS was first reported in the year 1981. So, this particular virus, they have an envelope which is made up of protein, and that envelope encloses the RNA as the genetic material. It is the genome of the virus. Now, how does the infection happen? Let's just say by any other means, retrovirus has entered the body. What does it do? It starts to infect the animal cells such as macrophages, such as helper T cell. And you know that whenever a virus causes any infection, it incorporates its genetic material, not any other thing. So the genetic material of the virus is incorporated into the cell of the animal. Now, this viral RNA synthesizes viral DNA using that enzyme called as reverse transcriptase. Now, since it's DNA, our genome is DNA. So now this viral RNA, it gets incorporated. It sort of gets mixed or aligned with the this thing, the DNA of the host, the genome of the host. And now the host cell, instead of synthesizing its own particle, it synthesizes the viral RNA and acts like an HIV factor. Right? So this is what HIV does. Now it goes to the macrophage, it goes to the T lymphocytes, and then it starts destroying it. And thereby, the number of T lymphocytes decrease in the body. And you people know that T lymphocytes, they help the B lymphocytes to do their work, to produce antibodies. So when these T lymphocytes are reduced, the immune system is severely compromised. So if a person has AIDS disease, so their immune system gets so severely compromised that they're not able to fight any even simple kind of pathogen also they can't fight. They can't fight common cold. Like the basic normal diseases are also the immune system is incapable to fight with. And then a person dies because of that. Now remember one thing, students, that the disease AIDS is incurable. There's no such cure available anywhere to treat the AIDS. Only the symptoms could be treated so as to relieve the person, but this disease is fatal. So the diagnostic test, just like how there was a diagnostic test for typhoid, same way there's a diagnostic test for AIDS as well, and that is ELISA, which stands for enzyme linked immunosorbent assay. That's what this test stands for. Now, the most cruel thing about this particular infection is that like a person has had, you can say, the HIV in their body. Now, furthermore, what will happen is this HIV will not let, you can say, any kind of symptom show. Yes, are you asking that this class is for 1 hour?
Yes, ma'am.
It is for 1. So talking about this thing, so like by the time the person shows any symptoms, by that time the particular pathogen has increased a lot in the body. Usually the showing of the symptoms appears 5 to 10 years after the infection. Okay. Then to more information that the World AIDS Day is like it's celebrated or made aware on the 1st December by the National AIDS Control Organization. In this day, on this day, the awareness about AIDS and everything is spread to people. Now the question comes along, how does it get transmitted? So first, sexual contact with the infected person, that can be the case. Transfusion of contaminated blood, right? If the person gets transfused with the wrong type of blood. Third, by sharing infected needles, such as when people do drug abusing, in that case, they take the drugs intravenously. So that is how another way of mode of transmission is there. And the worst one is that with if a mother is infected with HIV, she can transmit the disease to her fetus, to her child through the placenta. And always remember that HIV or the AIDS disease does not spread from touching, or anything of that sort, but it spreads only via bodily fluids. So AIDS is clear? Is it clear? Yes or no?
Yes, ma'am.
Ma'am,
Okay. All
ma'am,
right. Huh? Yes, Jyotita?
ma'am, what if a HIV person scratch like someone's hands? Will it spread?
What does the HIV person do?
No,
Scratches
if,
someone's
yeah,
head.
yeah,
No,
hand, like by mistakenly.
no, it will not spread like that. It only spreads with the exchange of body fluids. Okay, like during sexual contact, it will spread 100%. If infected, even if a tiny drop of blood of any other person infected with HIV enters the person of a healthy person's blood, then it will spread. No scratching, no biting, no slapping, nothing. Nothing will spread HIV. Understood?
Mm.
Okay, so this was the AIDS. The second disease that we have is cancer. Now, cancer is one such non-communicable disease, right? It does not spread from one person to another person. Now, this is the deadliest disease there is, and it is the reason of multiple deaths all over the world. If you think about India, so 11 lakh people every year. And if you think about the whole entire global scenario, then 10 million people around the globe. So talking about this particular disorder. So now see, you people know that there are two types of cell divisions in our body. What are they? Huh? Two types of cell divisions.
Mitosis and meiosis.
Yes, mitosis and meiosis. Now, if I ask you that what is this particular, like if I'm talking about the disease that is, so you tell me which type of cell division is used in the increase of number of cells.
Mitosis.
Mitosis. Very good. So when we talk about this particular disorder, so this disorder, like normally in our body, the cell division is very highly or severely regulated. That if there are many cells over here and our cells are divided. So as soon as they come in contact with the already existing cells, now the division will stop in that area. Okay, because there are cells already present. This property is called as contact inhibition. Now, contact inhibition is not found in cancerous cells, meaning they'll keep on dividing and dividing and dividing and will not stop even when they'll come in contact with other cells. So, then they'll start forming a mass of cell called as a tube. Okay, and this is a result of uncontrolled division. So in this case, what happens is that this tumor only causes the whole entire problem. The tumor is of two types, okay, depending upon whether it can spread or not. So benign tumor are those tumors that are localized. Localized meaning that they are found only in one spot and they'll not spread from that spot to other spots. So that is a benign tumor. Whereas when we talk about a malignant tumor, so this is the tumor that is, you can say if it is present in one area, then it will start spreading to other cells it come in contact. And that property of the spreading of tumor is called as metastasis, which is the most feared property of cancer or any tumor. So when we talk about a benign tumor, it can be surgically removed and it is harmless. It will never come back. But if the benign tumor is in a weird location, like in the spine, in the brain, then it is difficult to extract it out. It may lead to death, but the thing is that surgery can treat the benign tumor. Secondly, there's malignant tumor. So these tumors, like even if you remove them surgically, they'll keep appearing back. Like they'll keep happening and happening and happening. Now the question comes along, how is the cancer caused? The cancer is caused with, or you can say by the agents called as carcinogens. Now carcinogens, they can be physical, chemical or biological. So physical carcinogens would be rays, okay, radiations like X-ray, gamma ray, UV ray. They cause mutation and mutation causes the cells to have uncontrolled division. Chemical carcinogens are the ones such as tobacco, such as the smoke. So they can also cause or they can also trigger the normal cells to become cancerous cells. Lastly, there's the biological carcinogens such as in our body, in everyone's body, there are proto-oncogenes present. Proto-oncogene means these are normally inactive. It's just there, it doesn't get activated. But under the influence of the carcinogens, the oncogenes become activated and thereby the whole entire facade starts. Then comes what is the diagnosis or like the detection of cancer. So first is biopsy. It's surgically a part of tissue is removed to check it for tumor, to check it for uncontrolled cell division. So that is histopathological study. Histology is the study of tissues. Histopathology is the study of any disease in the tissue. Then there is radiography technique. Radiations can be used to detect cancer, like X-ray. Then there's CT scan, that is computed tomography. In this case, a 3D image of the internal organs is created and thereby the tumor is located. Then comes magnetic resource imaging, the MRI. You people might have heard about these tests. So what they do is that they use strong magnetic field to detect any kind of abnormalities that are there in the living tissues. Also, molecular biology techniques are also there, like PCR, like ELISA. Some of these techniques are there which can help in the detection and diagnosis of cancer. Now, the question comes along that what are the treatments? So first is the surgical removal of the benign tumor. Second, radiotherapy. Radiotherapy is when radiations are sent to kill that cancerous cell. And lastly, there's chemotherapy, wherein certain chemical drugs or chemotherapeutics are used to kill the cancers. But imagine it, students, if you're sending something via blood, it will reach all the cells, isn't it? Other than reaching the tumor cells, it will also reach the other cells of our body. Thereby, it destroys many healthy cells also. This is a downside of this treatment. So, there are a lot of chemical drugs that have a side effect such as hair loss, nausea, appetite, and weight loss. Clear, students? And lastly, if you remember, there were the cellular interferons, which are proteins that help the non-infected cells to be protected. So those are also provided, or those are also given in order to develop immunity. Is it clear, students?
Yes, ma'am.
Any doubts? All right, now you people note down, then we'll move forward. We have to start from autoimmunity, right? Tell me once you're done, I'll scroll it.
That
understand? Shall I scroll down?
Yes, ma'am.
So once you're done, tell me that I can.
Done, ma'am.
All right, students. So let's just keep it till here only for today's class. We'll continue in the next class. Okay. Okay, students.